Hi, this is a quick review of the blue-white selection. I recognize that I kind of rushed through it a little bit at the end of lecture 9 because I was concerned about not getting through it in the class time. Always a mistake to rush. And I did have a couple of questions, and I thought perhaps some of you probably know about it already and have already, get most of it. But if it's new to you or you didn't feel it was that clear in the lecture, hopefully this little intro will help a little bit. So here's the background for blue-white screening. So the plasmids that we use for blue and white screening, these are ones that have been specially engineered. So it's not like they're naturally occurring. None of the plasmids that we use are naturally occurring. They've all been heavily modified to serve certain purposes. And the idea was in the past, before blue-white selection, it was really hard to figure out which of your colonies contained an insert, and you'd have to make preps for all, a large number of them, and sometimes very few of them actually had your insert. And so this is a way of doing what we call screening, and I'll talk about screening versus selection probably at another time. So here's some key pieces of information that led to the development of the blue-white selection. So the gene LAC-Z, which has been very intensively studied because the lac operon is kind of classic gene regulation subject, and so it's well understood. And it encodes an enzyme called beta-galactosidase. Normally what this enzyme does is it helps the cell break down lactose sugar. If there's no other sugar available and there is lactose, then the genes get turned on and that gets broken down to make energy. And it was determined that you could basically plate cells that are expressing LAC-Z on a media with a substrate called XGAL. It's a gigantic long name, which I will give you at some point, but you only have to remember XGAL. And what it will do is it will modify it will break the molecule into galactose, or similar to galactose, and then the part that it removes is converted into a product that happens to be blue in the presence of this XGAL. So your beta-gal enzyme does something can do something it's not actually meant to do in the original cell. If you give it a different substrate than the one it normally works on, you can see this blue color. And so you have a visual, we call it readout, a way of seeing which cells have an intact beta-gal enzyme. Now it turns out, this is not true of all proteins, but the beta-gal protein can be divided into two pieces. One of which is called the alpha fragment, and the other one, I always call it the beta fragment, but I don't actually think that's the correct name for it. But the idea is you have two separate pieces, and they can be transcribed and translated separately and make two separate bits. And if you have just one of them or the other, you don't get a reaction when you combine them with this XGAL. However, if the cell has both of those two pieces, the proteins can kind of find each other and bind. Not all proteins will do this, but beta-gal is one that does. And then when they're together, they can still go ahead and make this catalysis. They can still make this blue product. So these are all the pieces of information that were used to then design a vector, a plasmid, that would allow us to use this feature of how beta-gal works in order to do a selection process. So we can just look at the colonies and know which ones probably have the insert and which ones are probably what we call empty vector. They're just the blue script plasmid and they don't have any insert. And there's a fair number of these usually in our experiments as well. Okay, so basically how do we use this information? First of all, we have to use bacterial strain. So the cells we use can only make part of the beta-galactosidase protein. So the first 126 nucleotides or 42 amino acids are not present. So this is just the partial protein. And then we have a plasmid, and it's called P blue script. There's other ones, but these days this was the first one, and this is the one we're using. And this one makes the other part. This makes the first 126 nucleotides And so the protein is the first 42 amino acids, and this one is called the alpha fragment. And so if you have a cell and it's this kind of bacterium and it has this deleted gene that makes a partial protein, but we put a plasmid in there and the plasmid gives it the other piece of the protein, now these two can still bind to each other, and they will still give us this blue product. Okay, so that's if there's no insert. So what we've done is the multi-cloning site, the place where the insert gets cloned into, is in the middle of that LAC-Z gene that's on the plasmid. So this is the plasmid. PBS is P blue script. And so basically, if you clone your insert in there, you still have the LAC-Z alpha fragment DNA, but it's interrupted by, in our case, either around 600 or around 1,000 nucleotides of sequence that is the wrong sequence. And so this can still be transcribed and we'll get an RNA, but most of the RNA is going to be the wrong sequence, and therefore we're not going to get any alpha fragment. This is going to be missing, and therefore the beta-gal fragment by itself, which this should have had a symbol, sorry about that, it's not able to make the reaction, and so we're going to get white colonies. Okay, so hopefully that's clear. The insert is right in the middle of this this little. It's quite a short gene. It's only, there's a little bit of extra sequence, but the coding part is only 126 nucleotides long. And if we put 1,000 of the wrong nucleotides in the middle of 126 correct nucleotides, there's almost zero chance of getting a protein that's going to do this job, right? It's just not going to work. Okay, so this just shows you, for those of you who are interested, this is kind of what the reaction looks like. This is X-gal, and this is the beta-gal protein when it's fully functional, and it makes a cleavage, but it cleaves two of them and sort of glues together two bits, these two pieces here, two like that, and those part are blue. Okay. Yeah, and so when we do our ligation, here we have our vector. It's been cloned with a restriction enzyme. In this case, it's showing you sticky ends, but it could be blunt. It doesn't really matter. And the yellow part here, that's the LAC-Z gene. So just so you can see the schematic, because this is a more professional looking than what I drew. And here's your insert DNA, right? You combine those in a ligation. Sometimes your vector just closes again. If these are compatible ends, then the vector can close. And if you've cut it with a restriction enzyme, the enzyme isn't going to cut every single molecule in the reaction. It'll cut most of them, but there might still be some molecules that haven't been cut with a restriction enzyme. So there will be a certain proportion in your reaction of vector plasmid that doesn't have the DNA you're trying to clone, and there will be a proportion. There's things we can do to increase the frequency of successful cloning, and we'll talk about those in lecture 10, which will be next Wednesday. So the ones that are carrying the insert are the ones that are going to have this gene interrupted, and now this protein is not functional, doesn't do its job, right? And so this is called screening. Selection is what we do with the fact that there's an antibiotic resistance gene. That's what the green is on this picture here. So we plate these on antibiotic, and we've done selection because only the ones that have the plasmid in them are going to grow, right? So we killed off all the ones that we didn't want to check out. And then we do what is called screening. We look at the colonies we got, and we use the color as a way of telling us, here's one that probably has an insert, and here's one that probably has what, as I said, what we call empty vector. It's probably contains well, it does contain a plasmid because of the antibiotic resistance, but it's very unlikely that this one has an insert, right? And this is just an image that shows what that looks like. And you can go to, I know that these are advertising vehicles, but a lot of the companies, they have quite good graphics, and they have very nice explanations of some of the procedures that I think for some students can really be helpful, because they make an effort to make the diagrams really clear and really straightforward to understand. And they also have written descriptions of certain procedures and stuff that are helpful. And they often have little how-to videos that are very helpful as well. So, you know, there's nothing wrong with looking at sites from some of the companies like Thermo Fisher and Invitrogen and so on. Not because I'm pushing for them necessarily for you're not going to buy stuff from them, but they certainly have sometimes really useful graphics and that sort of thing that can just help you understand a procedure a bit better. And this is another site. ResearchGate is great because they have these articles and they have quite good graphics and explanation and so on as well. And this is just another, you know, image of a set of results, and you know, you can see very clearly the blue colonies and the white colonies. And when we're doing our experiments, we're going to want to find, you can't see that very well, but the one thing we worry about when we see a lot of white colonies, especially around the edge of the plate, is sometimes the X-gal isn't spread very evenly. And so the colony might just have it might have a functional LAC-Z gene, but if it doesn't touch the X-gal, it can't turn anything blue, right? And so the colonies we really like are these ones. They're right beside blue ones. And so it's a good chance that the X-gal was well spread there. And so we have some, you know, some good colonies. We don't want to pick one that's touching another colony, because then we'll have some of the cells from the other one too. But finding a nice, pretty well separated one that's, you know, pretty close to some blue ones is helpful for us to feel more confident. It's always about trusting the results and feeling more confident about them. And I think that's probably enough, because there is some intro. There is an intro. It's quite an old one actually, but it's still totally relevant because we're doing the exact same thing in the lab for transformation. So the transformation lab videos will have more information as well.